And now we need to discuss few genuses of bacteria which are known as microaerophilic, means growing better in the decreased amount of oxygen, O2, and increased amount of CO2. First one is Campylobacter. It is a bacteria known by a causative agent of diarrheal disorders. In classification, it belongs to separate family, Campylobacteriaceae, and we have two main species, Campylobacter jejuni and Campylobacter coli. As it obvious from the name, they identified in different parts of intestine, and name is originated from anatomical location. During gram stain, bacteria is gram-negative, can be rod-shaped or comma-shaped, can resemble English S letter, or in some even literature sources it is described as a gull-wing shapes. Bacteria have polar flagellum and it is motile. It's usually moving as a corkscrew drive by rotation of the whole cell. Bacteria is non-sporing, and as we said, it is microaerophilic. So during different international investigations and studies, bacteria was identified as a leading reason of gastroenteritis worldwide, mainly in Northern America, and it is in number in top 4 in other different reasons of diarrheal diseases, usually E. coli, Salmonella, and some enterobacteria are taken first positions, and Campylobacter is fourth in that structure. So bacterial infection caused by Campylobacter is very difficult to differentiate, and usually both species causing almost similar presentations. The only confirmed factor of pathogenicity identified in Campylobacter is endotoxin of bacterial cell wall. It is lipopolysaccharide, and of course flagellum, as I mentioned already. So again, by clinical presentations, disease is the same as any other diarrheal disorder, manifest with common symptoms with intestinal disorders. As we said, bacteria should be cultivated in conditions with decreased amount of oxygen, some about 5%, and increased amount of CO2. And actually, bacteria can survive in up to 42 degrees Celsius, and it's preferred to cultivate bacteria at that temperature because other fecal pathogens will be inhibited and destroyed by that temperature, but Campylobacter will survive. We have specific culture medium for cultivation of Campylobacter. It's known as a Styrus medium or Campylobacter selective supplement, and it contains several antibiotics which can suppress normal flora of intestine, and Campylobacter is naturally resistant to it. Usually in that culture medium, bacteria will grow as a small, colorless or a pale gray colony with watery consistence and round and convex structure. So as you can see on the picture, culture medium have dark red, even brown color, and colonies are easily identified. They are very correct in shape, and bacteria could grow in the culture medium. So infection is usually transmitted from different types of animals or poultry, and transmitted, of course, through alimentary way by consumption of contaminated food and water, or in agriculture, even some cases of transmission of bacteria through contact with animals are described. So even without any consumption of food products or water, just simple contact with dirty hands after washing or playing with animals, bacteria can be accidentally engulfed by human and cause the disease. Usually, disease will last for several days and manifest with common symptoms, as we said, nausea, vomiting, abdominal pain, and diarrhea can be with the blood, but usually disease is self-limiting, and it will be spontaneously gone up to 1 week. In rare cases, so such complications as sepsis, and affection, damage of internal organs are described, especially significant complications, Guillain-Barré syndrome. It is a disease resembles poliomyelitis. By clinical presentations manifest with different types of paralytic disorders, but complications are quite rare in the case of Campylobacter diseases. So lethal outcomes are very rare and usually observed in young kids with not formed immunity or in elderly and immunodeficient patients with HIV infection or some other concomitant diseases with significantly suppressed immunity. As far as disease is self-limited, of course, antibiotics are not required, and usually immunity and normal flora will deal with bacteria itself, and only compensation of lost fluids and electrolytes is required to keep functions of internal organs stable. And of course, if you see that subject became worse, then monitoring of vital functions and some ultrasonic investigation of internal organs is required to exclude complications. But again, usually outcome and prognosis is very favorable. For prevention of the disease, of course, we don't have any specific measures, but common measures of food infections prevention are used, such as washing hands, keeping animals clean, disinfection, sterilization of water, and proper cooking of food products. Usually, it is enough to prevent any diarrheal disease and campylobacteriosis as well. Second, microaerophilic bacteria, which is more important and more popular nowadays, it is Helicobacter pylori or H. pylori. Bacteria, as it obvious from the name, have helical shape as a spiral, and pylori means that it is identified in pyloric part of the stomach. Bacteria as well as gram-negative and motile by polar flagella. Main number of properties are very similar with Campylobacter, and actually both bacteria are similar in structure and biological properties. And according to nowadays, the last study of World Health Organization indicated already that up to 75% of world's population are naturally infected with Helicobacter pylori. So this is and actually it's difficult, and we have nowadays two types of opinion in medical community and scientific community. Is it okay to have that bacteria? Maybe it is normal flora and nothing wrong with that bacteria, or maybe it is bad. And actually, till recent days, a lot of people thought that it is fine. You have Helicobacter pylori, it is your flora, nothing wrong with that microbe. You have nothing to be afraid of. But actually, nowadays, it is confirmed that bacteria is highly associated with so significant disorders as peptic ulcers and stomach cancer. But again, it's difficult to study pathogenesis of disease and any side of infection, because a lot of people naturally infected with that microorganism, but don't have any disorders. So during routine investigation and routine checkups, that bacteria is identified again in almost every 3 or 4 persons, but only few have some problems and some digestive disorders. So it is suspected that bacteria can be transmitted through unclean food and unclean water, or even during close contact between two persons, such as a kissing or sharing personal stuff. But again, it's not possible to confirm, because we don't have a group to compare with. Almost everyone is infected with that bacteria. It's just kind of normal thing. So mechanism of action of that bacteria as well is not clearly understood, but it is looking like bacteria, due to unique spiral shape, can as well move like a corkscrew drive, and it can invade epithelium of our stomach. It can pass through mucous coverage, and here it is hiding from our stomach acid. But constant and prolonged presence of that bacteria in the tissues of intestine will lead to some small damages, and stomach acid can pass through those passages and affect our mucous membrane. Initially, it will lead to gastritis, some slight disorder, slight uh, damage of epithelium. Later, it can progress to ulcer, and of course, ulcer is a significant damage. It is a direct damage of our cells. They will try to regenerate to restore the damage, and constant, continuous, and unstoppable multiplication of our cells with the time can lead to uncontrolled overgrowth, which is known as a cancer actually. So nowadays it is confirmed that bacteria is associated with those significant disorders. And actually we have still two positions. Some doctors say that it is obligate to kill bacteria if it is identified. But again, very often antimicrobial treatment, even correct, will not protect against further reinfection. And people who are treated now can be easily infected from common water sources or food products in 1 or few weeks after therapy is finished. So common idea nowadays that if you don't have any problem, any disorders and bacteria is identified, just regularly check your health, perform some ultrasonic investigation of stomach or common consultation in gastroenterologists. But just live with that bacteria. Be careful, but don't do anything. And if some problem will be identified, gastritis, uh, some any symptom, vomiting, nausea, without any obvious reason, and Helicobacter is identified, then you need to perform eradication of that bacteria. But treatment is prolonged. It's usually about a month, and it is a complex therapy combination of 2 or 3 antibiotics. Of course, it is sometimes very difficult. They can be toxic. Some of them can be expensive, and again, eradication of bacteria now will not prevent reinfection with the same bacteria tomorrow, because usually all family members share the same flora, and Helicobacter should be eradicated in the whole family in that case. So it is one rapid test for diagnosis of Helicobacter. It is known as a UDS test. We have multiple companies and multiple manufacturers producing that test, but idea is almost the same. Urease enzyme is unique enzyme identified in Helicobacter, and usually swab or some sample from teeth is sufficient to identify that bacteria. If it is present in intestine, enzyme is almost always identified in oral cavity. You need to take a swab from the teeth with a toothpick or any other thing, even some with the matches, and mix with the solution provided with the test strip. And if color of sample will be changed from initial to yellow, result is negative. From initial to red, result is positive. Actually, during practical activities, we usually perform the test in volunteer students. It is a simple test tube with reagent, urease reagent added. You need to put toothpick with the sample in the test tube. In 15 minutes, you see the result if color is yellow. No Helicobacter if it is red and Helicobacter is present. So again, the test can be done by mainly will of the subject, or if you want just to, if you're curious, do you have it or not, you can find the test in pharmacy office and buy and perform it in you. So it is all for lecture about anaerobic bacteria. See you in other lectures. Bye bye.